In this final section, we're going to discuss bulk transport and some more membrane protein functions. Sometimes cells move large objects or extracellular fluid rather than molecules or ions across the membrane. These processes use a lot of energy and rearrange sections of the plasma membrane. Types of bulk transport include endocytosis, which moves materials into the cell, endo meaning into, via three main processes. Pinocytosis is when samples of fluids surrounding the cell are brought into the cell. This is called cell sipping. Phagocytosis moves large objects into the cytoplasm. This is called cell eating because it's an object rather than a fluid. And then receptor-mediated endocytosis is when cell surface receptors bind specific large objects or molecules and then move them into the cell. All right, if we're not moving things into the cell via bulk transport, we move things out of the cell, and that's known as exocytosis, which moves material out of the cell and releases cell products into the extracellular environment. Exo meaning out of. Let's take a closer look at pinocytosis. Pinocytosis, or cell sipping, allows cells to take in samples of their environment. The plasma membrane wraps around small volumes of fluid and solutes. The cell membrane pinches together and a vesicle is released into the cytoplasm. So we can see that here, this sort of sipping of this sample of fluid that then pinches off from the cell membrane and creates this vesicle in the cytoplasm. The vesicle may then fuse with intracellular membranes, releasing the sample into cellular compartments. Phagocytosis, or cell eating, allows cells to take in larger objects from the extracellular environment. The plasma membrane surrounds an object, as you can see here. The cell membrane pinches together and a vesicle or vacuole containing the object is formed. Vesicles may then fuse with intracellular compartments, such as lysosomes, forming a phagolysosome in which ingested objects are digested. Receptor-mediated endocytosis. Receptor-mediated endocytosis allows cells to take in objects for which they have specific receptors. Receptors on the plasma membrane bind specific objects. You can see these receptors here binding these ligands. The receptors flow together in the plasma membrane and form a protein-coated pit, which you can see here. The pit surrounds the objects, the cell membrane pinches together, and a coated vesicle or vacuole containing the objects is formed, which you can see here. Vesicles may then fuse with intracellular compartments, such as lysosomes, forming a phagolysosome in which ingested objects are digested. Phagocytic cells. In the human body, phagocytosis is carried out by certain specialized white blood cells. They may phagocytose bacteria, dead cell debris, dust, and other foreign particles. Phagocytic cells are our garbage trucks. Phagocytosis is often the first step of an immune response or an inflammatory response. Heat, pain, redness, swelling due to injury. In this colorized photomicrograph, a macrophage is engulfing rod-shaped bacteria that you see here. Exocytosis. Exocytosis can be thought of as the reverse of phagocytosis. Cell products destined to function outside the cell must be released. Cell products, such as antibody proteins or hormones, are packed into vesicles by the Golgi apparatus, which you can see here. Vesicles fuse with the interior face of the plasma membrane here, and their membranes join the plasma membrane. Then cell products are released to the extracellular environment. How paramecia eat. Paramecia are unicellular organisms that share many characteristics with animal cells. Their eating and waste removal are examples of endocytosis, active transport, and exocytosis. Cilia, all around the outside here, sweep food particles into the oral groove on the plasma membrane. Vacuoles containing food are formed when the membrane pinches off from the cell mouth. Digestive enzymes are transported into the food vacuole. The resulting nutrients, such as amino acids and sugars, are transported from the vacuole into the cytoplasm. The remaining wastes are exocytosed via fusion with the anal pore. So you can see the endocytosis starting here at the oral groove and the exocytosis here at the anal pore. For advanced videos describing bulk transport processes, there are several links you can follow here. To view the entire library of advanced videos about membranes and transport, you can simply click the link here. More membrane protein functions. Proteins in the cell membrane serve additional functions beyond transport. Here are a couple. Defense. Antibodies and other cell surface receptors activate white blood cells to defend the organism against pathogens. Signal transduction. Receptor proteins receive signals from the environment, causing the cell to start or stop activities such as making new proteins, changing shape, or dividing. Cell-cell recognition. Cell surface glycoproteins bind to other cells' surface structures to communicate with other systems. Enzymatic activity. Biochemical reactions associated with membrane functions may be carried out by peripheral proteins. Cell adhesion. Connections between cells and between the cytoskeleton and extracellular matrix form tissues.